Brad, you've got something completely new here, a new way of looking at ovarian cancer. What have you been doing with this new agent? First of all, it's called trebaninib?
Trebaninib. Trebaninib is a new molecule that targets a new pathway. You've probably seen we have 7 phase 3 positive trials of angiogenesis in ovarian cancer, but they all target VEGF. And VEGF, as you know, has been associated with hypertension, bowel perforation, bleeding, thrombosis, wound healing problems, proteinuria, CNS disturbances even. So this study called TRINOVA-1, trebaninib in ovarian cancer, was to look at a new agent that binds angiopoietin 1 and 2.
And tell me about that.
Angiopoietin 1 and 2 is another angiogenesis pathway other than VEGF. And so we did a randomized phase 3 trial in recurrent ovarian cancer and showed a prolongation and progression-free survival with a hazard ratio of 0.66.
And what went before this? You've had some earlier studies.
So we had done a randomized phase 2, which showed a very interesting prolongation in PFS. Even in the phase 1 program, it was well tolerated and showed single-agent activity.
So you've got an extension of progression-free survival. The big question, of course, is overall survival. I believe you had what you call a trend, but that's always a little difficult to estimate.
So we thought that when we reported the PFS endpoint that it should be coordinated to at least half of the OS events. We wanted to tell you and the scientific community whether or not there was a decrement, maybe even a trend in improvement in OS. And we have a hazard ratio of 0.86 for overall survival. Again, only half of the OS events. But such a trend is very provocative, and I hope to share with you the OS next year.
Okay, so you've got positive results with progression-free survival. What about the big question, toxicity?
Of course. So we do not see the classic VEGF-associated toxicities, but we do see edema. Not only does angiopoietin 1 and 2 inhibit angiogenesis, but lymph angiogenesis. So the incidence of edema, generalized edema, localized edema, pleural effusions, ascites, weight gain, is 2 to 3 times what we see in the placebo arm. And remember, this was combined with weekly paclitaxel. So TRINOVA-1, weekly paclitaxel placebo, weekly paclitaxel plus trebaninib. Edema seems to be the real issue.
And what would you say to busy oncologists about managing that if this drug were to progress to licensing?
Yeah, so first of all, grade 3 and worse was only 2, 3, 4%, so not that serious. But grade 2 edema can still be very troublesome to the patients. So we use diuretics, we use compressions, elevation, and we need to learn more about the factors that predict edema and what the natural history of edema are.
What would you sum up them about the clinical recommendations, or the clinical implications anyway, future ones, so far?
So we need to combine it with other cytotoxics. We're combining it with pegylated liposomal doxorubicin in a study called TRINOVA-2. Remember, ours was TRINOVA-1. TRINOVA-3 is adding it to frontline. And I think because it works after anti-VEGF therapy failure, we want to combine it with anti-VEGF. If these are distinct targets, angiopoietin 1 and 2 and VEGF, and complementary, and in fact even mechanistically related to resistance, I think we want to combine anti-VEGF and anti-angiopoietin therapy together.
So what's the take-home message for cancer doctors at this point?
The take-home message is that angiogenesis is valid in ovarian cancer, and it's not all about VEGF. That we have a new target, angiopoietin 1 and 2, and a new agent, trebaninib, in inhibiting that axis.
And which particular patients can benefit potentially the most from this?
Yeah, so these were patients who had up to 3 prior lines of therapy with a platinum-free interval less than 12 months. So this is a large proportion of patients with recurrent ovarian cancer.
Brad, thanks very much.
My pleasure. Thanks, Peter.